Okay, good morning, everyone. This microphone is not for you to hear me better, it's because we are doing the streaming, and I will say a few words on that in a second. First of all, I would like to very warmly welcome you to the Bolin Days 2017. I'd like to welcome all of you. Given that it's only 15 minutes to 9, it's surprisingly many people that are here, so thanks for coming. I would also like to extend a very warm welcome to our ESAC, the External Science Advisory Group, that has probably traveled farthest to attend the Bolin Days. A warm welcome also to the SAG members. The SAG is the Science Advisory Group, and this is comprised of the research area leaders. The research area leaders will chair their sessions during these days. And I'd also like to welcome the board, but I don't see anyone from the board here yet, but I'm sure they will come. So my name is Nina Kirchner. I'm one of the co-directors of the Bolin Center. My other co-director is Alistair Skelton. He is sitting there. And we get fantastic help from Annika Bürström, who is somewhere else, but I will show you her a little later so you know that she is the magic person behind everything. So, we celebrate the Bolin Days in the honor of Bert Bolin. He passed away roughly 10 years ago, and you constitute the Bolin Center here, but institutional-wise, the Bolin Center is a cooperation between Stockholm University, between KTH and SMHI. And because we have some new ESAC members here, I go a little bit back in history. The Bolin Center started actually as a climate research school, and that was shortly followed, there comes the board, and that was shortly followed by a project that was called SUCLIM. And SUCLIM stands for Stockholm University's Climate Evolution, Variability and Sensitivity Project. That was funded for 10 years between 2006 and 2016 with roughly 120 million Swedish crowns. That is approximately 1.2 million crowns. This must be euros, right? This is translated into euros. Anyhow. And since then, we celebrate the Bolin Days. The Bolin Days started as simple annual meetings that were attended by much fewer persons than are here today. And then the Bolin Days actually evolved into a celebration of science. And this is a terminology that Alistair, I think, has coined. And now we celebrate science for 2 days. This year we have the 9th Bolin Days. It also means that next year there will be an anniversary. There will be the 10th Bolin Days. And we'll find something to make this a little bit extra special next year. So with Bert Bolin being behind the center and the idea of the center, I thought I put as a background one of these slides that you're maybe familiar with. We all now have these warmest years on records. These warmest years follow each other. And against this background, I thought we can also be a little bit proud of what we have achieved. We have consecutive records at the Bolin Days as well. Last year, we had the first Bolin Days as the new Bolin Center because we had merged with the EcoClim. And this year, we have, for the first time, a dedicated session from contributions from the human science area, and we have a complete external science advisory group attending the Bolin Days. So I think this is 2 very nice records in a row, and we will, as I said, find out what we do next year. If we put the Bolin Center in a larger frame and see what the global stakeholder events have been during the time when the Bolin Center was up and running, I've selected a few events here and put them on a time scale. Of course, in 2007, the Nobel Prize was awarded to the IPCC, and Bert Bolin was the founder, basically, of the IPCC. And then I checked here a couple of years. 2011 is when Sweden chaired the Arctic Council for 2 years until 2013. Then we had in 2012, we had the record low in the sea ice extent in the Arctic Ocean. In 2013, we had the presentation of the Working Group 1 Assessment Report 5 Summary for Policymakers that was hosted at the Royal Swedish Academy of Sciences in Stockholm. A year later, the United Nations had their open working group that proposed the 17 Sustainable Development Goals. And then in 2015, it was the COP21, as you all know, the Paris Agreement. And then in 2016, we had the Trump administration. And these are all events that, in some or the other way, have an impact on our work, because we work with climate science. And just very recently, COP23 had its final closing day. That was the conference in Bonn, 6th to 17th of November. And shortly thereafter, this was released in a press release as a state of the global climate in 2017. And it says that global ocean heat content is at record levels. Arctic and Antarctic sea ice is well below average. We have the highest September cyclonic index on record. We have the warmest 5 years on record. Sea level rise continues. Ocean acidification continues. Greenhouse gas concentrations continue to rise. And 2017 was also the warmest non-El Nino year on record. So there is lots to do for us in the Bolin Center to work with. It means that we need to continue what we are doing. We need to extend and disseminate the knowledge about the Earth climate system, climate impacting processes, climate modeling, human impact on climate, climate impact on ecosystems, biodiversity and human conditions, and how society can minimize the negative impacts. So in short, this is from our mission statement. We want to continue to carry out excellent research, graduate education, and outreach related to the Earth climate. And I've selected a few highlights from 2017. We are trying to do some housekeeping. This is not always easy because of technical challenges. We have a place on the Bolin Center website where we can put in all the publications. Not all of them are there for one reason or the other. I just went in there and checked. I found 14 publications in these really, really high impact journals. Maybe there are more, I don't know, but please let us know so we can include that. We do a lot of communication. We use all channels except the social media. We will be starting with that also, but I am not an expert on that, but we will get help on it. Alistair now has a Twitter account, so you can Twitter Alistair and he will hopefully reply. So we use all the classical media. We have radio, we have Bildningspodden, we have books, we have booklets, we have films, we have our Bolin Center Climate Festival. So we do a lot of communication, and this is very important that we really convey the results and the impact of our research to the community. Science is going to be continued. It's the same issue here. Annika sent you an email. We wanted to get some feedback how the funding harvest was, because the Swedish Research Councils have just decided on the funding. So according to what we know, our information may be incomplete. We brought home in the Bolin Center 7 grants from the Swedish Research Council, 7 from Formas, 1 Horizon 2020, and 1 from Vinnova. And this is quite a nice outcome, I guess, and shows that we actually have some momentum here going on. We will in the future try to compile an annual report so that we easily have information at hand where we can highlight events and also numbers that you can easily grab and that we have something. This will be forthcoming. We're preparing that hopefully very soon. And I think with that, I can close. So while you change your seats, so, very nice. Thank you very much. This microphone, as I said earlier, is not for you to hear me better. It's because we are doing an experiment. We are streaming lectures. Please let us know if you do not want your presentation to be streamed. But if you want, please use this because it goes directly into the computer and we can, this is why we use this microphone here. Otherwise, I'm very happy to hand over to the research area leaders. We will this year start in reverse order with the research area 8. We will be very strict in timekeeping in the interest of everybody. So first responsibility is yours to keep the time. And please also, at the end of the session, let people know what the next event is. So that really we want to try to get as many people in as many sessions as possible. And if you go out, please have a look at the activities that we have outside. And then I just hope that we really can enjoy 2 days of exciting science. And with that, this introduction is over, and I hand over to Sara.
Okay. Now it starts. Next. Yeah. I just need my presentation. Okay, I can say this. I'm Sara Cousins. I'm a professor in physical geography and one of the chair leaders of Research Area 8, Biodiversity and Climate, together with Karl Gotthard from Zoology and Johan Eilén from Deep. Right, we have a presentation. Excellent. I'm one of those that use a Twitter account and wanted Bolin to use it. So use hashtag Bolin Days if you're tweeting. So I will just start with our session by just presenting shortly the activities that we've done for 2017 and also upcoming events. So we gave seed money from RR8 to 6 projects. Several of them will present the research, also either as presentations or as posters. We're also recruiting new PhD students and postdocs. One is senior researcher, is it a postdoc or researcher in plant phenology, plant-animal interactions and climate. And I'm recruiting a PhD student to extreme weather effects on plant community composition. And Ayco, who's presenting later on, he's also recruiting a PhD student. We're also having we evaluating 15 proposals for new postdocs and PhD positions that we're working on at the moment. We had a workshop in April for the people that wanted to apply for money for PhD student and postdocs, and then we had a seminar in September by Susan Harrison from UC Davis in the United States, and we will have Jens Christian Svenning from Aarhus the 4th of December. I'm afraid I can't remember his talk, but he's working on big herbivores and climate change, primarily in the past, and rewilding. And then we will have a workshop in Tovetorp on the 6th and 17th of February. I know it clashes with another event, but we have invited speakers from the US and elsewhere, so we can't actually move it. So that's what we've been doing and are doing at the moment. And now we will have full presentations from PhD students and senior researchers. You can see that here, and I will keep shortly the time. I will sort of wave a flag 2 minutes before your time is up, hopefully before. So Olle is the first one to give a presentation.
How long do I have in total? 15. That's sans questions? That's with questions. Thank you. All right. So I'm Olle Lindestad, and I'm a PhD student working with Karl Gotthard. This is my study species, the speckled wood butterfly. And essentially what we're doing, let's see, does this work? Is we're interested in life cycle variation across space. So one way to think about this is that for almost any organism, at least in a temperate zone, but also elsewhere, there's part of the year, typically winter, that's unsuitable for reproduction, for most life events actually, except doing nothing. And then there's part of the year that's more suitable for mating, for growth, so on, foraging. And so if you imagine sort of a temporal distribution peak, for example, in this case of adult butterflies, you might want to time that so it falls within, or you expect selection to time it so that that peak falls within the favorable part of the season. And if we then imagine a spatial element to this, so that you have maybe a shorter growing season farther north, then you expect the shift, you expect a concomitant shift in the temporal distribution peak. You expect the phenology of this organism to be selected to change. And you can keep doing this sort of going north. And so this is what we mean by life cycle variation across space. And so, oh dear. Well, we expect always part of this life cycle variation to be plastic in other words, not genetically based, but just a passive effect of the environment. But some part of it we might expect to be genetic because we expect selection to push this phenology to follow the seasonal cycle. And so we're interested in sort of telling how much in a given system is plastic and how much is genetic, how much is actually due to intrinsic differences between the populations. And then if we further break down the genetic component, some of this will be due to actual selective events, so local adaptation we can call this. And some of this variation will be due to sort of more passive things like migration history or, so the genetic past of the population. And we're particularly interested in something that's mainly peculiar to insects, which is Voltinism. So a lot of insects will have multiple distinct distribution peaks of adults over the year, and this is actually different generations. So these guys are descendants, direct descendants. They're sons and daughters of these guys. These guys at this point are dead. So we have one generation of insects. They will give rise to offspring. They will then die shortly thereafter, and then the next generation will take over. And so you can have variation in this number of generations across space in an organism. For example, in the speckled wood butterfly. So what you see here is a map of southern Sweden, and these are isoclines representing season length. These are 10 days season length difference for each step here. And you can see that the Voltinism patterns, so number of generations per year is Voltinism. Univoltine if you have one generation per year. And this maps onto these isoclines fairly well. So how it works in the north is that you have a larva, and it grows, and it pupates, and then the pupa will go into winter diapause. This is another key word here. Diapause is a state of suppressed metabolism and suspended development and increased stress tolerance, in which most insects will spend the unfavorable season. But the way that it works in the south is that you have 2 generations per year, and this is intimately linked to diapause, because we have one generation here that will skip. So the first generation of growing larvae in the spring will skip diapause, and this is a decision. They know basically which generation they're in based on the day length. So they know what time of year it is by gauging how long the day is. And then they will decide, well, they will make we can call it a choice, to develop directly from pupa into adult butterfly. So the larvae make the choice, but the pupae suffer the consequences, you could say. Well, suffer. And then, of course, the next generation will have to go into diapause, and they will become the next generation of adults the next year. So you have one generation that diapauses, one that doesn't. All right, everyone following so far? So this is where it gets a bit complicated. So we expect as a result of this that there should be strong selection differences between these zones. There should be differences in what the optimal life cycle is. Part of this is just the critical day length at which you decide to go into diapause. And partly because the day length information that you get will be different across latitudes, and partly because you have to make a different decision based on that information if you want to achieve either type of life cycle. And for another thing, life history traits are expected to be different. If you want to have 2 generations in a year, I'm saying want, but you all know how evolution works here. If you want to have 2 generations per year, you have to grow twice as fast, essentially. So development time is one thing that we look at. All right, so to gauge how much of this is genetic and how much of this is plastic, we've done 2 controlled rearing experiments across 2 Voltinism clines, so one latitudinal and one within a latitude. This is mostly altitude. And these are our sampling locations, and so one of them is common for both clines. And the design is fairly simple. We get adult butterflies from the field. Let's see, do I have a timer? Yeah. And we get F1 offspring from them, so larvae. We rear them in controlled conditions where we manage their photoperiod, the day length. And then when they pupate, our main response variable is whether or not they went into diapause at a given day length. So the results of the latitudinal cline look something like this. So here we have 4 populations. We have 4 day length treatments. I should add here that this is actually a recent invasion, so these have any variation in this area, if we can find it, should be due to either, well, doesn't matter. This is actually what's important here. What you're looking at here is the pupil diapause response across day lengths. So our treatments are here, 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 and here. And then these are extrapolated or fitted probability curves of going into diapause. So over here, the day length is short. That means you're early in the season, or late in the season, sorry. They would interpret this as it's late in the season, where in the second generation, everyone needs to go into diapause. And indeed, you see basically full diapause response in all populations. And then, as you go into higher day lengths, it means you're earlier in the year. It's basically midsummer. And here we have individuals averting diapause. But the point at which this happens, the point at which they interpret it as being early or late in the season, is different for different populations. And so this indicates genetic differences between the populations. And you see that the biggest difference here is between the univoltine and the bivoltine populations. That's where you find the gap. So that's what you expect, where the strongest selective difference should be. And if we compile this with some early results from earlier studies, we get 6 populations. These have 2 generations per year. These have 1 generation per year. And you can see that it doesn't linearly follow latitude. There's sort of a break here. Right. So the second experiment is, I think it's a little more interesting, because here you always expect selection differences based on latitudes, because the information you get, as I said, is different. Days are longer in general when you go farther north. Here, these populations are all on the same latitude, more or less. They all more or less get the same day length information, but they still have different life cycles. So here you might ask how much of this is just due to temperature, because it is actually warmer on these islands here, Öland and Gotland, than it is in the South Swedish highlands up here. So here we also had 2 temperature treatments to account for this, to see if maybe they use temperature to interpret day length differently. But it turns out actually the main effect is still due to just intrinsic differences in these photoperiod thresholds between populations. So here we have 4 populations, but it looks like you have 3 lines here. That's because 2 populations, these islands, they behave exactly the same, which is notable because they have different, we know from genetic data that they probably have very different migration histories, which indicates that there's actually selection has molded them to be the same, even though they have different backgrounds. These are the 2 single generation per year populations. They fall up here. And temperature complicates things by shifting the response thresholds by about half an hour, but they do so the same for all populations. Right, so how much time do I have now? 5 minutes? That's great. So I have time to tell you about what we did with this information, which is construct a phenological model. So here basically we're interested in seeing if you were to, let's say, move populations between these locations, given that it is, well, so they have different thresholds, but doesn't matter, right? You always need to ask yourself whether the differences you find between populations actually have an effect. And so what we did was use these biological parameters, or use these results as parameters in a phenological model, where we fed in temperature profiles for the past 10 years for each of these locations. And we basically tested the parameters of each population in local and foreign conditions. And I probably can't explain exactly what it did because that'll be a little more involved, but plus these figures are unreadable. But this is just here to show that we did the work. And the results essentially say that no, population traits do affect Voltinism outcome. So if you swap population traits between the locations, you get erroneous results. Whereas if you let each population have its appropriate day length thresholds and so on, they produce exactly the phenological variation that we see in the observation data. And a key result here is that larval photoperiodism is important. So this is something I haven't mentioned so far, but this species is very photoperiodic in the larval stage. So not just what the pupa does, whether or not it diapauses, whether or not it goes into overwintering mode, but also how fast the larva grows. So these are the single population per year, univoltine northern populations. And here you see that basically the longer the season progresses, so the shorter the day length is, the faster they grow. If it's midsummer conditions, they basically take forever because it's adaptive. They can take it easy. And you see the same thing in the southern populations, except if day length is so long that it's adaptive for them to have a second generation. So if these individuals feel that they're in the first generation, and then they develop super, super fast. And at this point, it's basically temperature dependent. Over here, photoperiod actually overrides temperature. So you can have them at different temperatures in this, or actually, so long as they're below this threshold. So the first generation, or the second generation in the year, when they're past the point where they want to develop super fast to have a second generation, they know that they're in the diapause generation. They can take it easy, and then photoperiod will slow them right down until photoperiods get short enough that they need to hurry up, if that makes sense. So that's actually a really interesting result. You need to account for photoperiod because it overrides temperature, it seems like, in parts of the response range. Right, so in conclusion, we found large differences in life cycle traits across the Voltinism boundaries, as expected. There is a mismatch between migration history and population traits in certain places, which might indicate local adaptation. We're working on confirming or denying this genetically. Phenological modeling suggests that at least some of this Voltinism variation is actually upheld by population traits and not just environmental differences, climate differences between locations. And with that, I would like to thank some of these blacked out people and our funding sources, and thank you for listening. All right, then we can run a tight ship then.
Okay. So, hi everyone. My name is Eva. I'm a PhD student at the Stockholm University, and I work in the Department of Physical Geography. In my research, I work with dendroclimatology and dendroecology. So, dendroclimatology you may be familiar with. It's where you access past climate variability through tree rings. And since trees are living organisms, they also record the ecological conditions that are present at the time that these rings are formed. And then we have this dendroecology. And where is the... Here. So you can trace back insect outbreaks, forest competition, or even fire scars. So where is then a tree ring? So every year, a new ring is produced in a tree trunk. And as you can see here, this is an annual ring. If we take a closer look, we see that we can divide it into early wood and late wood. The early wood, it's formed in the beginning of the growing season, and the late wood towards the end of the growing season. And it's actually the differences between the early wood in density and the late wood that makes these sharp boundaries. In Scandinavia, trees grow from April to September. So, in my PhD project, I try to differentiate between climatic parameters and anthropogenic influences on this tree growth, especially in urban environments, because as dendroclimatologists, we tend to collect our trees as far as possible from human interference, right? But then we end up comparing and building up our regression models with climate data that is collected within these urban limits. And the land use change between these different landscapes can lead to up to 3 or 4 degrees in temperature, but also in water moisture because of this water runoff. And since we have different competences at our department, through cooperation with the Landscape Ecology Group, we hope to, with our data, contribute to a broader research scope. So we have then 2 goals. The first one is the impact of this land use change on the growth performance of these old trees. The second one is to study the effects of these land use changes on the time and dynamics of the forest expansion. So for this specific talk, our site is the Stockholm region. And the Stockholm region has one of the longest meteorological stations in the world. I think only 3 or 4 places have as far back as the Stockholm. And if you want to statistically back up your data, so nothing better than 250 years to calibrate and validate your models. So for this project, we got the seed money from the research area to go to Ixla and bleed. So the Landscape Ecology Group has been conducting studies and surveys in these two islands for a long period now. So we think for our further conclusions that we can take advantage of the data that they've been collecting. So our site criteria from the dendrochronological point of view, I would say the most important one is the type of trees. So we focused on coniferous because it's the most used tree in tree rings reconstructions. During the field campaign, we visit 15 sites based on these criteria. And what we do, the field methods, is that we use the Swedish increment borer to collect the tree cores, a GPS location for each tree, and we try to take a transect from the old forest into the regrown one. And this is the kind of disturbance that we are looking. So you have these individuals that are dominant, but then they start to compete for light and for nutrients. So what is the effect that this has on the tree growth? In the lab, we used the conservative tree ring width with the measuring table, but we are also using the iTrax multi-scan because you got an optical image and a radiographical image. And the radiographical image actually allows you to use different dendro softwares and makes possible to do different kind of analysis. And the kind of feature that we are looking here is this. So one ring is produced every year, and then you see here this kind of release that has some kind of stand dynamics disturbance. What divide them? You do this in different ways, but what I did, I produced three different chronologies. So the old stands, the intermediated ones, and the new trees. And then I plot them against each other. And what we are looking is this kind of feature here. So you see when the new trees start to grow, the old ones start to decrease their growth, right? But also an interesting feature is that if we look into the growth rates, these new trees are growing much more than the old ones, even when they were beginning their growth. So an additional outcome from this project was, while we were sampling one of our sites here, we took a walk into the forest just to have a look into these old stand forests. And we found these kind of trees. So pines that are growing in these very, very thin layers of soil. This soil is very poor. These trees are very stressed. And from the forestry point of view, they have no value at all, which is great for us, right? Because then we go there and we collect these trees. And we have here a 370 years old pine. And we found specimens with 400 years old. So remember what I say about these growth rates being different between these trees? So here we are sampling a 370 years old pine. And here we are sampling a 40 years old pine. So these trees are growing in this very rich soil that were former grasslands. So they have no competition, so they just grow. And we wanted to know is this data good enough to do a climatic reconstruction, or what can we do with it? Just a simple test. You may think the correlation is too low, but this is just 10 trees. And it's also like ringweed. We think with the radiographical images, we will be able to produce density, and the correlation will be much higher. So what is interesting here for you to see is that we have May. The trees grow from April to September, right? But we have no correlation with July. So what we think is these thin layers, these trees are very stressed by the temperature. So actually, when it gets too warm, these trees shut down their growth. And then you have a higher correlation for August and September when the temperatures decrease again and they get more moisture. And if we take a look now into one of these tree cores that we get from these radiographical images, can you see this feature here? So this is the annual ring growth, and what we think we see is what we call a false ring. So the tree starts to feel the stress from the hot, and then it starts to close their growth. But then the temperatures get mild in August and September, and then start to grow again. And if you see the differences in density, it's as much as big the late wood as it is the light wood. And this is common to, I have seen this feature in many of the tree cores that we collected. So now what we are doing is collecting samples from Tyresta National Park and also the Royal City Center National Park to see if this is a feature from only these trees in the archipelago, which are influenced by this huge water mass, or if it's actually, or if the inland trees from these sites also present the same kind of patterns. So expected results, this is an ongoing research, and we hope that we can produce some forest expansion maps by the end of this pilot project, also to have a better understanding of this human and forest interaction, to clarify a little bit of this bias issue that we had in these reconstructions. We now see the potential for a climatic reconstruction where we can go further back in time than 250 years from this observational data. But we hope by providing this dating accuracy from the dendrochronology that we can help the landscape ecology group to improve these estimates on community composition change rate. And that's all. Thank you.
Any questions?
Why did you choose to work with me? Because in dendroclimatology, this is the most used species for climatic reconstruction, and also because some species they produce false rings or they produce no ring at all, and also this is one of the longest living trees. When if you go to birch, they don't live longer than 100 and 150 years. So if you really want to go further back in time, it's good to have these deciduous trees.
Thank you. That was a nice presentation. But from the point of view of dendrochronology, for a biologist, this seems very expected that you have this complex life history, growth, how it's regulated with the seasons and so on. Does not that make it very difficult? If you could come up with a model even, you have to know the community composition and all these things in order to do good to estimate climate from these data, which is, of course, what a biologist would estimate in some way or expect. So is there a chance to somehow resolve, for instance, temperature from these kinds of data, where you have all the other influences of seasonality and competition and what kind of nutrients you have in the soil at that particular moment, depending on the previous history of land use and these kinds of things?
So what is your
You have the ambition to somehow from this model it so that you can for instance, estimate climate from these types of
Yeah,
data?
of course, that's what we want, to see the climate change also, and to see if we have differences in these trees that live in the archipelago and trees that live inland. And also we want to produce a kind of a transect, so we have been collecting trees from different places in Stockholm. But there's also, like, when we talk about dendroclimatology, trees have different, like in Sweden, if we go further north, the trees have a higher correlation with temperature, but the further south you go, they kind of have a mixed correlation between precipitation and temperature. So we are trying to get the key parameter that influences their growth. Is that the answer?
Okay, thank you. We will move on to Ayco.
Should I turn this on in any way? Okay. So yes. So my name is Ayco Tack, and I'm working as a big lecturer at the Department of Ecology, Environment and Plant Sciences. So today I wanted to talk about some new series of climate-related projects, and this is on the relationship between climate, life history, and a multitrophic food web on oak. So Olle, 2 talks ago, kindly introduced what's climate and life history. So let's start with what's an oak and what's a multitrophic food web on it. So here it is. So this is an oak tree, and an oak tree provides a habitat for a very diverse community of insects and microbes. So here you see just some of the lepidopteran species that associate with oak trees in Sweden. And some of the groups that are very easy to work with and I've been very interested in are, for example, leaf miner species, where the larvae of the insects is spending its whole life feeding inside of a single leaf. So here you see different species, some eat like straightforward, others form mines. And it's only the adults that move around. So these are really easy to count in the field in the end of the season. Another species I've become very interested in is this gray or whitish patches on the oak leaf. So in the autumn, many of the oak trees are really covered by this gray layer of mycelium. So it's an oak powdery mildew pathogen. So it grows on top of the leaf, and it's only its feeding organs that suck the energy or that's feeding on the epidermal cells. So I wanted to discuss 2 related questions in this presentation, and neither of them have really full data yet. So this is the first question. What is the impact of temperature on the phenology of the oak food web? As Olle explained, the phenology is the timing of events in nature. For example, bud burst and when a plant starts to flower. And I have some preliminary experiments from a field heating experiment and a growth chamber experiment. The very related question is, what is the impact of temperature not just on the phenology, but on the number of generations insects have within a food web? And as you all know now, the number of generations an insect has within a season is called Voltinism. So this is really just a set of conceptual ideas. So what may happen with phenology in a food web? So this was a recent review by Thackeray et al., where they looked at how sensitive different trophic levels are to changes in climate. So they made predictions for 3 trophic levels: primary producers, primary consumers, and secondary consumers for 3 emission scenarios. So you can see that the primary producers were expected to advance by 2 or 3 days, primary consumers by 6 days, and secondary consumers by 3 days again. So you can imagine that this different sensitivity of different trophic levels would somehow have an effect on the synchrony between the primary consumer and the primary producer, the secondary consumer and the primary consumer. Just to place it within the oak food web, so the primary producer I will talk about is the oak tree. The primary consumer are the insect herbivores, plant pathogens, and also endophytes. And the secondary consumers are the parasitoids. Parasitoids are insects that eat other insects, either from sucking them from the inside out or from the outside. So this is the first experiment. So for this, we designed two different cages, or two different treatments. One is the control treatment, and then the warming treatment. So when I talk about we, it really is Maria, who's sitting here, and Adam from SLU in Uppsala, who did all the work. And within these cages, we placed replicate 3 individuals belonging to 5 different genotypes. So this is how it looked in reality. So above you see the different cages. There you see Maria again. And here you see the infrared heaters that we use to warm the cages. So here I should point out, those were funded by the Bolin Center. So if you're ever interested in using these infrared heaters, you should talk to me. But I should warn you that if you use them, you're really significantly contributing to climate change by the energy consumption. So what happens with trees in spring when you start heating them? So these are some of the very first results. So on the y-axis there, you see the different phenological stages you can observe in spring. So that basically goes from bud break to the full development of the leaves. This y-axis doesn't span that exact range, but and each line here is the response of one genotype to the warming. So I think you can see a few things here, and this is the only statistically supported result I'm going to show today. So warming accelerates tree phenology, and tree genotypes seem to differ in the spring phenology, especially in the control treatment. There seems to be quite some variation among the different genotypes. And maybe there's an interaction between the two. So different genotypes respond differently to temperature. So we have a lot of research on spring phenology, but I also am interested like what happens actually in the autumn when a lot of insects are feeding on the oak trees. And it's quite important for the later generations to actually finish their life cycle. So here's the first result on leaf senescence in the end of September. Basically, the y-axis ranges from low values that are that represent yellow leaves to green leaves, higher up the y-axis. And again, you see the response of the different genotypes. So I think, just based on the figure, that leaf senescence seems to depend both on a warming treatment and the tree genotype. So there's some kind of different response by the different tree genotypes to warming. We also looked at leaf drop later in the season. And we didn't analyze or plot that yet, but this is how it looked like. So in the warming treatment, I think it was mid-October somewhere, the trees still had for a fair amount their leaves, whereas in the control treatment, the trees all had dropped their leaves. So I think based on the figure alone, we can already conclude that leaf drop is strongly affected by the temperature. So what about the insects and the microbes living on the trees? So here's a figure for the oak powdery mildew, which you see on the top, which is nice because you can observe it straight in the field. And then we also looked at the endophytes, so the bacteria and the fungi that live in the leaf, but they don't cause any disease symptoms. So for the latter, you have to take leaf discs, and then you have to metabarcode the bacterial and the fungal community. So that's work in progress. But here's the first graph on the pathogen infection level. And without having done any analysis, it seems at least that there's the counterintuitive result that infection is lower when you start warming the trees. That's quite puzzling, actually. So from this experiment, we don't have any results yet from the insects. But we did another experiment with insects and phenology, which was done by Adam, which was a growth chamber experiment. So this slide is a bit mixed, but you can still read the question. What's the relative importance of insect genetic variation in temperature and the timing of spring emergence of herbivores and the parasitoids? So basically what we did in autumn is to sample leaf miners and gollers, 2 types of insects on oak, along a gradient going from France to Finland. So we collected thousands of insects. Again, when I use we, it's actually Adam and Maria also. And we stored these insects during winter and spring in 5 different climate chambers. And the temperature in the climate chambers reflected the temperature at the sampling points. That's why I call it reciprocal. And then you can look, when do the different insects start to emerge? So that's a lot of work. And here's the results for one single species. And this shows that the individuals of this particular leaf miner species, then when you collect them from France or from the Netherlands, they need more degree days before they actually emerge. So the conclusion preliminary is there seems to be genetic variation in emergence times. So individuals from different origins hatch at different degree days. Here's one graph where there's both this leaf miner and its peristoid. And now we focus on the different growth chambers. So you can see that in the growth chamber with the climate from France, it's the leaf miner that hatches first and then the peristoid. Whereas in the other treatments, it's the peristoid that emerges first and then the leaf miner. So I think there's some indication that climate affects the synchrony between the leaf miner and its peristoid. You're not waiting yet, right? Great. Then I'll talk a little bit about the second question, which is very related. What is the impact of temperature on the number of generations of insects? And this actually comes about from the fact that in the field you actually observe that some species in some years, at some places, start to make second generations. And in some cases, they just cannot finish that generation. So I think they're all doomed. So I've always been wondering, what's the effect of starting a new generation on the population dynamics? So here's a little bit the conceptual idea. So you have spatial and temporal variation in climate, as Olle nicely has been talking about. That, as Olle showed, has an effect on the Voltinism. There's also a trend through time. This is a paper by Altermat that showed that after the 1980s, it's more common to find second and later generations of butterflies and moths than it was before the 1980s. So climate change may really be changing the number of generations of insects. And this really has ecological and evolutionary consequences. It may increase the speed of population growth and also the speed of adaptation. And what I am interested in is how this would affect food webs. For example, a herbivore may start a new generation, whereas the peristoid doesn't follow. And I think that we have studied Voltinism pretty well for single species. Many new questions arise in the context of food webs. So this brings me to the new project from the Bolin Center, which is a collaboration with Philip Lehmann here and Karl Gotthard both from zoology, who know much more about Voltinism than I do. So the project exists out of 4 different questions. So the first question is, what is the spatial and temporal variation in Voltinism? So here you see the example for a single leaf miner species. You see that in Sweden it has an abundance peak before midsummer and one after midsummer. So I think the underlying reason is that this species, in some parts of Sweden and in some years, it has a second generation. But it may become more common with an increase in climate. The second question is, what are the drivers of Voltinism? Some species in a food web may be affected by the spring temperature, others by the summer temperature. There will always be species in the food web that do not change the number of generations. So it's fixed, genetically fixed. The third question is, how does Voltinism affect resource selection and performance? So imagine you're a species that usually has 1 generation. If you start a second generation, the quality of the oak leaves in the autumn must be so different than the quality of the oak leaves in the spring. So you're faced with a totally different resource quality. And this will necessarily affect the performance of the species and the choice of the oak leaves. And it will also affect co-occurrence patterns among different species. So the fourth question is about the synchrony between the different trophic levels, where I may just point out one scenario, and that's that if a herbivore starts an extra generation, and normally its population is controlled by a natural enemy, but the natural enemy does not start a new generation at the same time, then it can have very fast population growth. So this may actually lead to insect outbreaks. So there's my 2 minutes left. So here are some take-home messages. I think that climate change may profoundly shape plant-based food webs through changes in phenology and Voltinism. But this really is work in progress. So thanks for listening. And I guess you should.
Thank you for an interesting presentation. For your fourth research question, are you targeting any specific species that depend on the lower trophic levels for that research question?
What do you mean with lower trophic?
Well, if you go back one slide, this is not my area of expertise. So the wormy guys are the ones that you're looking at, right?
Yes.
And so there must be some higher trophic levels that depend on the wormy guys coming out at a specific time. What species are those, and are you looking at those as well?
So yeah, some of the species I'll be looking at are the leaf miner species. So they live their whole life inside of a single leaf as a caterpillar, or as a larvae, I should say. And they're particularly easy to work with because you can just collect them. You know, you just collect the leaf and you get the adult out of it. And in many cases, you get the parasitoid out of it, the natural enemy. And then I hope that we can also work with some free-feeding insects. So yeah, we will mostly caterpillars, so moths, I think.
And they're dependent on these species. What's dependent on these species? I'm missing the dependencies.
And they're being attacked by parasitoids.
Okay.
Do you know what's a parasitoid?
Yes.
Yeah.
Yes, that I do.
Yeah. So parasitoids lay eggs on top of larvae, either inside of the larvae. In that case, you get a larvae living in a larvae, and it eats it from the inside out. But sometimes it lays the egg also on top of the larvae. In that case, it kind of sucks it out from the outside. But in this food web, then you also have still insects that lay their eggs inside of insects, So those are hyperparasitoids. So it's quite a complex food chain, and it's quite fascinating to think. So one other scenario here is actually that what happens when the herbivore doesn't create a second generation, but the parasitoid actually does create a second generation, then it doesn't have a host to live in. So in one case, the parasitoid would just die, and if it massively forms a second generation, it would have a strong effect on the population growth. Alternatively, it goes to a different host. So many of the parasitoids, they prefer one host, but they can feed on multiple hosts. So in that case, the population dynamics of this species and that species will be coupled for the parasitoid, which you often refer to as a parent competition.
Okay, I'm afraid we'll have to take questions with a coffee break. There are plenty of spaces up here.
The old ecoclim, and it's sorry, that was the wrong one. It's done by, where's the pointer? No, this one. Anyway, there's lots of people involved, and most of the really tough work has been done by the people in Boulder, Diana, Tana, and Jose. And we have been exploring this particular question in a system of a butterfly and its host plants. And much of the thinking around the project started out with data like this, collected in a review by Camille Parmesan, who's here in the audience. This is pretty early work, showing basically that there are massive changes in phenology in many organisms. And the typical pattern is that, as you see, they are negative here. Spring phenology becomes earlier. That's the typical pattern. Another interesting pattern from this graph is that these changes are not equal for different types of organisms. So, for example, which is particularly interesting for us, butterflies seems to change more quickly than the plants they feed on. And this may, of course, lead to changes in the synchrony between these groups of organisms, and that might affect how fitness and interaction patterns of these species. And if we think about it, why would we try to find a model for understanding why we expect increasing synchrony with changes in climate? One way of thinking about it, and the way used, is this: that so we think different organisms have what we call different reaction norms, which means that a reaction norm is how a particular genetic category has some change in the phenotype, in this case development, in relation to an environmental variable such as temperature. And in this particular case, we can think of A and B as different species, and they, if they are dependent on a particular kind of synchrony, you can see that if the temperature is changing, this type of synchrony might change and might be detrimental to this interaction, and maybe, if it's very important for any of these species, extinction of the species, or at least from particular areas. However, a perhaps more common thing is that a given species A here is not interacting in this particular process only with one species, but with several. And these different species might all differ in these thermal reaction norms. And the main effect of this kind of situation, if there is a change in temperature, might not be that species A disappears or has very much lower fitness, but instead it changes the way it interacts, or which species it is interacting with. So the interaction patterns or intensities might change. And this was what we were trying to investigate in this project, using this particular butterfly, the orange-tipped butterfly, Anthocerus carominus, who feeds on multiple host plants of the Brassicaceae family. And it has a particular phenology or life cycle, this butterfly, in that it lays its eggs on the flowers on these host plants, and the larvae are feeding on the flowers and the fruits of these plants. And since these plants typically only flower during a relatively short period in spring, the butterfly has to time its life cycle to its host plants. And another thing that's important for the system is that the butterfly has only one generation throughout its range, and it overwinters in the pupal stage. So the processes partly during winter, which I will not talk about now, but also what happens with pupal development in spring, and how it has to time its pupal development to the host plant development in spring. And we did this in a series of different, throughout a latitudinal gradient in Sweden, from southern Sweden, the Stockholm area, and northern, or at least in Ångermanland, where we did field work during several years. And we also did lots of experiments where we bring plants and animals from these locations to the lab and do experiments in climate cabins and climate chambers. Experiments that investigate the degree how temperature affect developmental synchrony between these species, and also how the phenology of the plants affect the position preferences of the butterfly. And one of these lab experiments was relatively recently published. So maybe I need this. Which one is the röda? Ah, there it is. Okay, so thanks. So the results here, this is the results of this growth chamber experiment, where we basically have the butterflies, pupae, that has been overwintering, and the overwintering plants. We bring them to the climate chamber and estimate their developmental rates in a set of different temperatures. And here you have the 3 different locations where they come from in the columns here. The rows show basically just 2 different plant developmental events. This is development to the first flower, and this part is development to the fruit stage, the first fruit. In all these graphs, you see thermal reaction norms estimated from the data, which you don't see because it just becomes too messy in the graphs. The red lines is the thermal reaction norm of the butterfly, of the pupae. That is the time it takes from we start the experiment until it hatches as an adult. Whereas the other colors are different of the 5 of its different host plants. And these stars here denote the cases where we find a significant difference in the slope, in how they react to temperature between the butterfly and the plants. And overall, you can see that, of course, very commonly, as all organisms here develop faster in warmer conditions, and that there are very few stars, so that the butterfly seems to be very well synchronized throughout this temperature range with the development of its host plants. So it's doing well. So a rise in temperature might not have a big effect in the synchrony of these species in the south. There are, however, some examples where the plants react more quickly, or their development increases more rapidly than the butterfly in warmer temperatures. And especially here in this northern population, it appears that we have more examples of the potential of increasing temperatures creating some kind of change in the synchrony between these species, between the butterfly and its host plants. Okay, and the second part of this experiment was doing an opposition experiment, where we let the females choose among host plants indoors in conditions like this, where we vary the phenological state of the host plants. So there are some young, some old host plants, and we wanted to investigate basically, does some kind of temporal mismatch between the butterfly and the hosts in fact matter for the female butterflies when they oviposit? And what we find is, what they found was basically, yes, it does. So here you have an estimate of a phenological index, where 0, quite non-intuitively, is old individuals. They only have seed pods, they only have fruits, no flowers left. Whereas 1 here means these are individuals that yet don't have any flowers, they only have buds. And then the females are allowed to choose among plants that different phenological index. And what we find is this sort of hump-shaped optimal phenology, where it appears that the butterfly females like plants that are in this phenological 0.6, which means basically that 60% of the flowers are open. There is also some effects of the species identity, but they are kind of difficult to understand really, and very weak. So the conclusion here is that phenology is much more important than the variation between the different species. So yes, one conclusion we can take is that a shift in synchrony is perhaps not so very likely, except in the northern population, but if it happens, it is likely to influence the interaction patterns. So as I said, we also did field work in these 3 populations, where Diana, Tenna, and Jose and field assistants scored the phenology of thousands of plants and also checked whether they had been oviposited on, whether there was an egg laid on them. And this was done over 4 years. And well, we don't have to care about that. So this is simply to show that we did these field trips during a time when the butterflies had started to lay their eggs. So we could really estimate the butterfly choices. And these are some of the results from these 4 years of hard labor. And here's a kind of a complicated graph. So on the y-axis, you have host use, relative host use, which means that in a proportion, which means that this point here, in this year, about 60% of the eggs in this region was laid on this particular host plant. And the take-home message here is basically that, and on the x-axis, we have the deviation of every of these plant populations from what we estimated to be the optimal oviposition, the optimal phenological state where the butterflies like to lay their eggs on. The negative values mean here that these plants are much older than what the butterflies like, whereas these plants over here are younger than the butterflies like. And 0 is right on the money, so this is what they like. And what we find is that obviously it appears that the phenological state of the host plants is very important for their host choices. But of course, it is also the fact that we cannot really tease apart the effect of host species and phenology here, since host species obviously have different phenologies. But it appears at least that what we saw in the lab experiment also occurs in the field. And I'll just skip this. This is for good reasons, because the next part of interactions is when the eggs are laid, the larvae start growing and are then in a developmental race where they're host plants, where the larvae needs to finish their growth before the host plants spread their seeds. And there's strong effects on butterfly fitness, on these from phenological index and the temperature where this interaction happens. I'm sorry, I'm a bit late here, so I'll have to go to the conclusions. Yeah, and now I have half a minute. Okay, good. So the conclusions so far from this study is that the butterfly is relatively well adapted to retain its synchrony with its host plants in a range of temperatures. Although in certain areas, increasing temperature has the capacity to change spring synchrony. As both oil position preference and larval performance are influenced by the relative timing of the interacting species, temperature-induced changes in synchrony may indeed lead to changes in interaction patterns. And in this type of non-specialist herbivores, the degree changes in relative timing with increased temperatures may indeed lead to host shifts rather than direct fitness benefits, fitness costs. Although then, of course, there might be indirect fitness costs because some of the hosts that they now use are suboptimal compared to the ones they used before. And the final conclusion is that in these types of species that are well adapted to temperate regions, and the seasonality that we see here might in fact be relatively well buffered against changes in climate, because they are already well adapted to this huge interannual variation in the weather that we see. And with that, I'd like to again highlight Diana, Tana, and Jose, who's done almost all the hard work, and Bolin and Sandra, who worked on the same system in slightly different adjacent projects, and some of us others who tried to help as much as we could. And the funding was primarily EcoClim, to some degree the Wallenberg Stiftelse, and of course also the Bolin Center this year. So, thank you.